All right, this lecture is going to be on the general and special senses. For general senses, we're going to be pretty brief. It mainly refers to sensory receptors found throughout the skin and underneath the skin. And then special senses is going to require the bulk of our time here. So we're going to start with a little background. So general senses are your senses to respond to things such as temperature, pain, stretch, or pressure. Special senses are highly specialized, like taste, smell, vision, hearing, and equilibrium, which is your state of balance. So receptors are structures in the body that respond to stimuli, like a Pacinian corpuscle is a receptor you've seen before. So for this part right here, each receptor has its own receptive field, and that's just the area that it responds to. So if you look at this picture, you can see they have two receptors, one here, and this receptor has a smaller receptive field than this one. And so depending on the receptor, the receptive field size just varies. The other issue that I want to bring up is tonic versus phasic receptors. So if you look at this picture here, see each little star is a stimulus, like maybe just a tack hitting your finger or something that you need to respond to. So stimulus, 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 and this is the response. So you can see with a tonic receptor, you get the same response every time. If you compare that to a phasic receptor, you still get 3 stimuli, but the response actually gets smaller the more and more stimuli there are. So it kind of, the receptor kind of adapts a little bit. And so depending on what part of the body you're talking about, some parts of the body have a lot of tonic receptors, which are good. Then some parts of the body have a lot of phasic receptors, and that's good in certain aspects also. The classification of receptors, you can divide them into general versus special senses. And we'll basically talk in detail about the special senses, so there's not a whole lot else to say right here. But the other thing is just modality of stimulus. So chemoreceptors respond to stimuli. Respond to chemical stimuli. Thermoreceptors respond to temperature. Photoreceptors respond to light. Mechanoreceptors respond to mechanical stimuli like stretch. Baroreceptors respond to pressure. And nociceptors respond to pain. So be familiar with what each of those receptors responds to. As far as the types of receptors that we have, so they can be unencapsulated but with no connective tissue, or they have connective tissue around them. To be honest, it doesn't matter for our purposes. So I just wanted to bring up some different ones, and some of these you've seen before. So free nerve endings we saw when we talked about the skin. Hair root plexuses we talked about when we talked about the skin also in hair. You can see this is a good picture of the nerves surrounding a hair follicle. Merkel disc we've seen before, a little disc sensory receptor found in the skin. And then 2 of these you've seen before. The Pacinian corpuscle, you know very well, responds to pressure and vibration. Meissner corpuscle, found on the dermal papillae, respond to touch. And then we have these other ones, Ruffini corpuscles, which responds to like skin distortion, and then also Krauss bulbs, which respond to really light pressure. So just kind of be familiar with the different types of general receptors that we have. And that's pretty much it for the general senses. So the rest of this is going to focus on the special senses. So we're going to start with taste. So taste is called gustation, and we have taste buds called gustatory receptors. They're found in these structures on the tongue called papillae of the tongue. So there are 4 types of papillae that are found on the tongue. So filiform, fungiform, valate, sometimes called circumvallate, and foliate. So kind of running through these, filiform kind of look like little fireballs. So they're short and spiked. These are interesting because these are the only papillae of the tongue that do not have taste buds. Fungiform are more block-like, like this, and they have taste buds. You can see these little taste buds right here on the side and the top, so that responds to taste. Then we have one that we usually see a lot in class because we have a lot of microscope slides on it, valate papillae. or circumvallate papillae. Either one is correct. They form a V on the back of the tongue, and they're loaded with taste buds. So you see all these taste buds on the side. And then lastly, foliate taste buds on the side and found on back and sides of the tongue. So let me just go back to that picture of the tongue. And these are the vallate or circumvallate papillae. So they form this nice little V on the back of the tongue. All right, taste buds. So they're basically, a taste bud is a collection of cells. So it has these supporting cells surrounded by gustatory cells, which actually sense the taste. At the tips, which would be on the surface of your tongue, you have these little microvilli. These are what actually respond to taste. So microvilli, gustatory microvilli respond to taste. And then when they sense it, that stimulates these neurons, and the action potentials are transmitted back towards the brain to let you know what something tastes like. So a given taste bud will last about 7 to 10 days, and then they're replaced, which is good. So it allows us to kind of constantly maintain a pretty good sense of taste. All right, so the sensations that you can taste: salty, sweet, sour, bitter, and umami. So umami is the one that most people are not familiar with. It's described as a savory or meaty type flavor. Sometimes it's just described as delicious. So that's the other kind of taste that we don't really hear about that much, but we've all had it at some point, I'm sure. I put in yellow sour and bitter because those are the ones that are not very pleasant. And if you think about it, it's just kind of it's not 100%, but most things that are not good for us to put into our body don't taste good. So they're sour or bitter. Think about if you tried to drink gasoline. It wouldn't taste salty, it wouldn't taste sweet, it wouldn't taste delicious. It would taste bad. Sour, bitter, however, I've never tasted it, so it's got to be one of those. But it's kind of a protective mechanism that we have. So a lot of things that aren't good for us just don't taste good. There's a couple exceptions to that, but nevertheless, that's a relatively common thing that you'll see. And then the gustatory pathway, you can see cranial nerve 9 and cranial nerve 7 conduct a different, like we talked about with the cranial nerves, different parts of the tongue. For different parts of the tongue, we have these 2 different cranial nerves that help transmit the sensations of taste. So we're going to move on to vision. So vision is a rather intense sense that we have. So starting with the main organs we're going to be dealing with are obviously the eyes. And so we'll go through the anatomy of the eye soon enough. But just the accessory structures we need to talk about first. So the eyelid, which closes your eye. Eyebrow up here. Eyelash. These are all there to protect the eye. So all these accessory structures on the outside are there to protect the eye. The eye is a very critical organ that we have. Other accessory structures, we have tarsal glands that secrete this oily secretion. So located here, and thought to prevent tears from getting too excessive. And then the conjunctiva, which lubricates the eye, are these things right here. So this is good to know because a lot of times this will get inflamed, and it's called conjunctivitis, or sometimes called pink eye. And it can have different causes, bacterial or viral, but conjunctivitis is pink eye, and that's what's inflamed, the conjunctiva. All right, lacrimal apparatus. Lacrimal refers to tears. So this is the basic setup of how tears flow through across the eye and then eventually into the nasal cavity. So you have lacrimal glands out here above the orbit. They secrete tears. The tears are going to generally flow across the eye from lateral to medial direction. Go through these tiny little canals called lacrimal canaliculi, eventually into a lacrimal sac, nasolacrimal duct, and then end up in the nasal cavity. So this explains why if you have a lot of tears, you're crying, you oftentimes get a runny nose because these excessive tears kind of just all drain. Some of them obviously come out of your eye onto your face, but a lot of them drain into the nasal cavity. So looking at the anatomy of the eye, so 80% of the eye is in the eye socket or orbit. There's two big sections. There's two big cavities. There's anterior and posterior. So looking at this eye, you're looking at a section that they cut like a sagittal section through the eye. So you're looking like a side view. This is the front. This is the back. So this person would be looking this direction, and they did a sagittal cut right through the eye. So this structure right here is called the lens. We'll get to it soon enough. But everything in front of the lens is called the anterior eye cavity. Everything behind the lens is called the posterior eye cavity. And the posterior eye cavity is going to be important for us because it's also called the vitreous cavity or vitreous chamber. It contains this gel-like fluid called vitreous humor. You'll see a picture of it, but it's loaded into the posterior cavity. It kind of keeps the eye under certain pressure. And if you cut into an eye, like normally we would do an eye dissection, it'll all kind of just burst out. So if you like took a scalpel and punctured the eye, it's under a little bit of pressure, so a lot of times it'll shoot out. So that's the vitreous humor inside this vitreous cavity or vitreous chamber. All right, so the layers of the eye wall include the fibrous layer, which is the outermost, the middle layer, which is called the vascular layer or tunic, and then the inner layer, which is called the neural tunic. We're going to go through them in that order and talk about the different parts of the eye and what they do. So starting with the fibrous tunic, there's two big parts to it. So in the front, you have basically what you'd be looking at the front of your eye, this clear structure. This is called the cornea. And so it is there to kind of protect from things from getting into the deeper parts of the eye, but also it kind of bends the light. So light passes through it and it changes the direction of the light a little bit. So that's the cornea right in front. A lot of times people with a abnormality called an astigmatism, the cornea is just misshaped. And so the light rays get sent somewhere else in the eye and it gives them blurry vision. So they can correct this if you're a candidate for it. That's where the LASIK surgery comes in, but usually it's just reshaping the cornea if they can do it. Everything else, the white stuff that you see, which we see mainly the white in front of the eye, but it actually goes all the way around, that's called the sclera. So the sclera is the fibrous white tissue of the eye. It's dense, irregular connective tissue. All right, moving into the middle layer, the vascular tunic. So a few things going on here. I'm going to go out of order just to kind of make it a little bit easier. But this structure right here that is colored, it's a colored ring, a colored disc. That's called the iris. And in the middle of that disc, there's basically an opening. And it appears as like a black dot. That's your pupil. But it's not actually a black dot. It's actually an opening. And that allows light to get in. So it looks black because you can't see anything behind it. But that black hole is the pupil. The iris can change size to basically make the pupil dilated or constricted. And so there's a muscle called the dilator pupillae muscle, which dilates the pupil. And then sphincter pupillae muscle constricts it. So I'll show you that on the next slide. Other parts of the vascular tunic. So all this layer with the blood vessels in it, this is just called the choroid. It's loaded with blood vessels. And the other thing that's really important here is this. This is called the ciliary body. And you just got to envision that it does go all the way around. So it kind of looks like a pleated structure. But it has smooth muscle. And you can see that smooth muscle is attached with these little ligaments going to the lens. And so this ciliary body is going to be useful in changing the shape of the lens by either increasing or decreasing the tension on these ligaments. And as you're going to see soon enough, that's going to allow you to focus when you look at something. So ciliary body, which has smooth muscle, choroid, which is all these blood vessels, and then the iris, which is the colored disc. All right, and this is what I was talking about. The sphincter papillae muscle constricts the pupil. Dilator papillae muscle dilates the pupil. Notice that the sympathetic nervous system, which you know about, dilates the pupil, and the parasympathetic constricts it. So normally in class we would do a quick little experiment, like when you shine light in somebody's eye, what happens to the pupil? And the reflex is going to be that it constricts the pupil as a protective mechanism. If you're lying in a dark room, your pupils are going to dilate to try to let more light in so you can see a little bit. So a good example of this might be if you were in a room with no windows and all of a sudden you turn the lights out. For the first several seconds, you're not going to be able to see anything. It's just going to look black. But as time goes on, you might be able to see a little bit and make out things. That's because your pupils are starting to dilate a little bit and let more light in. All right, and lastly, the inner layer is called the retina, or I'm sorry, the neural tunic, and the big part of it is called the retina. And this is where we have the photoreceptors that are going to allow us to see. So this is it, and I know this is kind of hard to envision what we're looking at, so you're just going to have to envision. I'll try to go back so you can see this picture. So if you think of this as the eye, light's going to come through here, and it's going to hit this back wall. This back wall is the neural layer. All right, so this is what we're about to talk about, this back wall right here. It's made up of something called the retina. All right, so with that being said, I'm going back to this picture. So here we are. Here's the back wall. Light's going to come hit it. So if we took a little section of this back wall, it would look like this. And so light would be coming in from this direction, hitting this neural layer. So all of this is the neural layer with the photoreceptors and some other cells. So the inner layer has photoreceptors and neurons, and this is where all the action is as far as transmitting these light rays into action potentials to tell your brain what you've seen. So what's going to happen is, let me just go back and go to a bigger picture. So light's going to come in this direction. It's going to hit these cells right here. These are photoreceptors. There's two types. There's rods and there's cones. So we have a lot more rods than we have cones. So all the yellow ones are rods, the green ones are cones. These photoreceptors, like we said earlier, detect light. So when light comes in, they're stimulated, and they're going to generate action potentials. That action potential is going to be transmitted to these next neurons here. These are bipolar neurons. We'll just call them bipolar cells. And they continue the impulse. And you remember bipolar neurons, one of the things we said about them, they're very rare. And this is one of the few places we have them, in the retina. And then from the bipolar cells, there's these other cells called horizontal cells, which mainly do a supporting role. The action potential is going to be transmitted to these, what we call ganglion cells. And the ganglion cells are going to continue to conduct that action potential towards the brain, and you can see how they're all kind of merging together. They're going to form what we're going to call the optic nerve. All right, and these other amacrine cells here, not overly important, just play a supporting role also. So the big things are, you have the photoreceptors, rods and cones. That transmits the impulse to the bipolar neurons, which transmits the impulse to the ganglion cells, which form the optic nerve. One other thing about the rods and cones, the cones are important in color vision and high intensity or bright light, and the rods are basically everything else. So if you were colorblind, the issue is with the cones, not the rods. So rods, more dim light, black and white vision; cones, color vision, bright light. All right, so another interesting concept here is, like, this is that same picture of the back of the eye. So you might think that the light is just hitting all over the place, which it does, but ideally you want the light to hit right here, this little spot. This area is called the macula lutea. There's a little indention right here called the fovea centralis. This is where we have the best vision. So if you don't have a lot of light hitting here, your vision's not going to be great. So ideally this is where most of the light is hitting. This is a good area because it's loaded with cones, and and so it gives us the best vision. The other thing is, if you look right here, so these, where I'm putting the mouse right now, these are the rods and cones, this little layer right here. So where these, let me just backtrack a little bit to make sure we understand what we're talking about. So if light came in, it would hit maybe these photoreceptors right here, rods and cones, goes back through the bipolar neurons into the ganglion cells. The ganglion cells are going to transmit the impulse back here and form the optic nerve, which is going to the brain. All right, so if you follow this layer of photoreceptors, the optic nerve cuts through right here. So you have a section right here where the optic nerve is, where there's no photoreceptors. So if light were to hit right here, you wouldn't see anything. And usually, because you have two functioning eyes, you don't ever notice it. But there's an experiment you can do. It's in your lab book if you want to look at it. And you hold basically a sheet of paper with an X and an O on it, and you cover one eye, and basically you focus on one of them, but keep the other in your peripheral vision. Well, the one in your peripheral vision will just disappear at some distance from your eye. So it's a fun experiment to do. So if you want to look that experiment up, it's in your lab book under one of the vision labs. But key point, where the optic nerve is, it's called the optic disc. There's no photoreceptors. That gives us what we call is our blind spot. All right, back to the lens. So remember we had the ciliary body that had muscles, and from that they had these little suspensory ligaments that attached to the lens. So when the ciliary muscles relax, they push up and away from the lens, which increases the tension. The lens gets flat. That's what you do when you want to look at something far away. And then when they contract inward, so it slackens or loosens the ligaments, and that decreases the tension. The lens becomes more round, and that's what you do when you look at something close up. So you may notice when you're looking at something really close, you feel like a little bit of stress on your eye, versus looking at something far away. That could explain it. But really quickly, if you want to, like if you were holding your hand in front of your face, you could focus on your hand, but outside, like in your peripheral vision, you might see something across the room. If you're focused on your hand, what's across the room would be blurry, right? But then if you focus what was across the room, then your hand would be blurry in your peripheral vision. So that quickly, just like in a split second, you can change the shape of the lens so you can focus on something else. And that's how we can focus on things close and far away really rapidly. All right, as far as the visual pathway goes, pretty simple. So yeah, the photoreceptors, bipolar neurons, ganglion cells, optic nerve, which goes into the brain. You have the optic chiasm, which you've seen before, crossing here. And then you don't have to know all this, but most of the visual stimuli is going to the occipital lobe of the brain.